Hello, everyone. Today we're continuing our deep dive of Richard Dawkins and Yan Wong's book, The Ancestor's Tale. In this episode, we're going to discuss the formation and function of coral reefs. So let's jump right in. In the previous installment of this series, we met the bizarre cnidarian parasites, staurozoans, cubozoans, jellyfish, and hydrozoans, including the siphonophores. These cover one of the two main branches of the phylum Cnidaria. In this episode, we're going to travel down the remaining branch. Here we encounter the corals, anemones, and sea pens. Recall that jellyfish transform from a zygote to a little flat planula. That planula roams around until it has found a suitable environment. It metamorphoses into a polyp. The polyp undergoes strobilation, and then juvenile medusae, called ephyra, emerge from the strobila. The life cycle of corals is identical, but only up to the polyp stage. That's where it ends for them. There is no strobila or medusa. Indeed, the polyp was originally the final stage of the cnidarian ancestors. Since the polyp is their final stage, corals, anemones, and sea pens are sessile during most of their life cycle, although some are still able to make a swift getaway whenever the situation calls for it. There are three main clades we're going to cover today: Octocorallia, Seriantharia, and Hexacorallia. Collectively, they form the subphylum Anthozoa. The name essentially translates to flower animals due to their vague resemblance to flowers. There is also one genus of buttercup called anemone, Greek for daughter of the wind. Indeed, sea anemones are named for this flower. After reaching the polyp stage, anthozoans have tubular-shaped bodies with an orifice on top surrounded by tentacles. Basically like a tubular jellyfish, but upside down. That might seem a bit odd considering the strange shape of, say, the brain coral or a sea pin, but who said each anthozoan consisted of only one polyp? One coral consists of numerous tiny polyps that can be organized into any number of shapes. The sea pin looks like a feather, but each barb of the feather is lined with polyps. On some corals, such as the aptly named elegance coral, Catulophylla jardinii, the individual polyps are readily visible. All of the polyps of one coral are connected via a shared digestive tract called the gastrovascular canal system, similar to the situation in the siphonophores mentioned in the previous episode. Unlike the siphonophores, though, coral polyps are more or less identical in shape and function, except in the case of sea pens. Tissue called the cenosarc connects each polyp and overlies the calcium carbonate skeleton, which it secretes, also called the cenosteum. When the coral dies, all that is left of it is the mineralized skeleton. Historically, the skeletal material has been referred to as the coral, and the animal that produces it is the polyp, or Polypifer, hence the name of today's tale. Each new polyp is formed through asexual reproduction, i.e. budding, but when creating a new coral colony, gametes are released to the currents. At this point, you might be wondering, what is the difference between a coral, an anemone, and a sea pen? Well, first, let's go back to the three main clades of Anthozoa: Octocorallia, Seriantharia, and Hexacorallia. It would be very convenient for us if these clades corresponded neatly to our folk taxonomy of corals, anemones, and sea pens, but alas, phylogenetics does not work like that. What we call corals are widely distributed within the Octocorallia and Hexacorallia, while sea pens and anemones are deeply nested in each, respectively. Additionally, there is also the genus Millepora, which are called fire corals due to their burning stings, which can last for days or even weeks. But these are not bona fide corals since they have a medusa stage. They are hydrozoans, closely related to hydras and siphonophores. Returning to the Anthozoans, members of Octocorallia have polyps with eightfold symmetry, while members of Hexacorallia have polyps with, as you might guess, sixfold symmetry. Octocorallia contains the blue coral Heliopora cerulea, which is the only known octocoral that builds massive skeletons made of aragonite, but most octocorallians are known as soft corals. Although almost all of them possess calcareous skeletal elements called sclerites, and some secrete a tough protein called gorgonin, such as the Venus fan Gorgonia flabellum. Sea pens of the order Pennatulaceae also belong within the Octocorallia. Sea pens are filter feeders found on the seafloor globally at many depths. Unlike other corals, their polyps are anatomically specialized for specific functions similar to, but not as extreme as, siphonophores. One polyp loses its tentacles and becomes very large, forming the base and central rachis of the sea pen, while many tiny polyps branch off from it, with some that take in water, the siphonozoids, and others that catch and consume food, the autozoids. Now let's move on to the other two groups, Hexacorallia and Seriantharia. These two are sister groups more closely related to each other than either are to the aforementioned Octocorallia. Seriantharia is often included within Hexacorallia as a basal lineage, but that depends on how broadly you define Hexacorallia. The relationships still remain the same regardless. Let's treat them as separate for our purposes. Hexacorallia contains 5 orders: Actiniaria, the anemones; Zoantharia, the zoanthids; Antipatharia, the black corals; Corallimorpharia, which are called the false corals because that's not confusing; and lastly, Scleractinia, the stony corals with skeletons made of aragonite and are the major reef builders of the world. Then there is Seriantharia, also called the tube-dwelling anemones. These are bilaterally symmetric, and their tube is secreted by a specialized organelle called the tychocyst, which is structurally related to the stinging nematocyst. Even though these are called tube-dwelling anemones, they are only distantly related to the aforementioned actiniarian anemones within Hexacorallia. But then why do we call both of these anemones? While corals are colonial animals made of many tiny polyps, anemones are solitary, living as one large polyp. The fact that two distantly related groups are solitary suggests that the ancestral Anthozoan was also a solitary polyp, and colonial forms evolved multiple times independently. Remember that all cnidarians have cnidocytes, hence the phylum name, and we are typically most familiar with the nematocyst-type cnidocyte, in which the secretory cnidocyst ejects a harpoon to spear prey or inject venom. There are other types of cnidocysts, though, such as the spirocyst, that releases a tiny lasso. Fascinatingly, when a team of researchers knocked out the transcription factor NVSOX2 in the anemone Nematostella vectensis, the stinging nematocyst was transformed into the lassoing spirocyst, indicating that the spirocyst is the ancestral cnidocyst that has been adapted for new functions over time. Transformations like this are called homeoses, which we discussed in the fruit fly's tale. These are extremely common, such as the antennapedia mutation, where insect antennae become legs. The reason for this is that a section of a body is triggered by a homeotic signal to develop into one specific organ. When the correct homeotic gene isn't functioning properly, the identity of the segment changes. The developing of the segment still continues, but it falls towards a sort of default state. In this case, the expression of antennapedia induces the development of the antenna, but without antennapedia, legs, the default state, develop instead. This also informs us about the evolutionary connections between these organs. Antennae are derived from walking limbs, as can be seen in the much more limb-like antenna of Cambrian arthropods like Fuxianhuia. Returning to our cnidarians, corals are well known for forming aggregates that can range from just a few meters to over 2,000 kilometers, over 1,200 miles, such as the Great Barrier Reef. They can exist from warm equatorial regions to the cold depths off the northwestern coast of Scotland, called Darwin's Mounds. This area has sand volcanoes where pressurized water bursts upwards through sand, creating a volcano-looking structure. Here, the deep-sea coral Lophelia pertusa uses sand as its substrate, unlike everywhere else it is known to grow, and the mounds also support populations of the coral-like giant foraminifera Syringamina. This is a member of the foram clade Xenophilophoria, which are present in the deep sea across the planet. Because of the large maze-like shape of xenophyophores, numerous animals can hide and live within them, such as isopods and copepods, sipunculid and polychaete worms, nematodes, brittle stars, and mollusks. Xenophyophores are a lesson in miniature for our tale today. Corals have been around for a long time, going all the way back to the Cambrian. However, Cambrian reefs were not formed by corals, they were instead formed by cyanobacteria, calcareous algae, and ancient sponge relatives called archaeocyathids. The archaeocyathids went extinct in the Middle Cambrian, caused by the Sinsk anoxic extinction around 513 million years ago. Although not one of the big five mass extinctions, it was the first major extinction event of the Phanerozoic. This event also divided up the Cambrian radiation in two phases, characterized by the dominance of stem groups in the first phase and the dominance of crown groups in the second phase. The first reef-building corals were hexacorals of the orders Tabulata and Rugosa. These appeared in the Ordovician, however both went extinct during the Permian-Triassic mass extinction. The modern group of reef-building corals did not appear until the mid-Triassic. From the late Jurassic to the late Cretaceous, rudist bivalves, which looked a bit like trash cans, became major reef builders, but they died out during the end Cretaceous mass extinction. Though seafaring people have long known of coral reefs, how exactly coral reefs became atolls, which are ring-shaped coral reefs, was unknown until 1842. Geologist Charles Lyell, the father of uniformitarianism, proposed that coral atolls were superficial incrustations atop volcanoes that lay just below the water's surface. As Darwin circumnavigated the globe from 1831 to 1836, he read the first edition of Lyell's 1830 book, Principles of Geology, and took much of it to heart. While in Chile in 1835, Darwin witnessed an earthquake that raised a mussel bed out of the water, indicating that land can indeed rise and fall, just as Lyell said. However, when Darwin read Lyell's explanation for atolls, he was unconvinced. Fortunately, Darwin was provided with the opportunity to test Lyell's idea when he arrived on the Cocos, or Keeling, Islands in the Indian Ocean in April 1836. Darwin knew that corals are found mostly just below the water's surface in the sunlight or photic zone, i.e. down to 30 meters. But what if instead of the atoll being just the reef at the very top of a volcano, the entire atoll is made of coral with the remnant volcano below? Just like all other cnidarians, coral planulae recruit in favorable environments, and their environment has to be in an area accessible by lots of sunlight. That means the planulae can only recruit in shallow waters on the sides of land masses. As the aging volcano erodes and/or subsides, new corals must recruit atop the older corals to stay in the optimal water conditions. The end result is that the living corals stand atop a mountain of skeletons. Darwin tested this idea by taking progressively deeper samples of the Cocos Islands, vindicating his idea, and then he later published his results in his 1842 book, The Structure and Distribution of Coral Reefs. Lyell was so impressed by Darwin's conclusions that he even changed his mind. And subsequent studies have shown with some modifications, like the addition of plate tectonic theory, that Darwin was right. So why do corals typically need to stay in such sunny zones? This is due to their endosymbiotic dinoflagellates of the genus Symbiodinium. Understand that cnidarians have ecological relationships with various other organisms. For example, the parasitoid ciliate Vampirophrya pelagica uses cnidarians as a secondary host, digesting their tissues after being passed from copepods. By contrast, the anemone Edwardsiella lineata infects the ctenophore Nemiopsis ladii, and once inside, it metamorphoses from a planula into a vermiform parasite. Once the parasite kills its host, it reverts to a planula and exits. It may become a polyp next, or infect another ctenophore. Trematodes, cestodes, and nematodes are known to infect cnidarians, and pycnogonids, or sea spiders, have been observed hitching rides on jellyfish. Some sea spiders consume the tentacles of jellyfish, while others simply use them as vehicles, possibly for catching prey. Similar behavior is observed in argonaut octopuses. They attach to a jellyfish, capturing zooplankton as it drifts. And many fish hide under the subumbrella to avoid being eaten by larger predators. The symbiosis with Symbiodinium, by contrast, is mutually beneficial for both the host and the endosymbiont. For this reason, the association has popped up multiple times in corals and anemones, and even in the upside-down jellyfish Cassiopeia that we discussed in the brine shrimp's tale, the Papuan jellyfish, whom we met in the previous tale, and the thimble jellyfish, Lanucha unguiculata, who is the most common cause of seagoer rashes in the western Atlantic Ocean. The reason is that Symbiodinium passes products of photosynthesis like glycerol and glucose to the host, while the host passes waste products like nitrogen and phosphorus to the endosymbiont. The cnidarian acquires its endosymbiont from the environment, which are common throughout tropical waters, having the greatest diversity in the area between northern Australia and southeastern Asia. Because of their usefulness, Symbiodinium are harbored by a number of other organisms, including sponges, flatworms, giant clams, and even some large foraminifera. So, since Symbiodinium requires photosynthesis to produce its exports, corals housing these tiny organisms must live in the photic zone. This association allows coral reefs to be the oasis in a desert ocean, as they have been called. In other words, the waters around coral reefs are oligotrophic or nutrient poor, but the algal coral symbiosis allows both to thrive in these regions. But Symbiodinium isn't the only member of the coral hologenome. Bacteria, archaea, fungi, and other protozoans and viruses play roles too. Gamma proteobacteria and alpha proteobacteria, actinobacteria, flavobacteria, and cyanobacteria are often present, although the species diversity of these clades can vary with coral species, biogeography, environmental conditions, and even life history stages. One very important gamma proteobacteria is Endozoicomonas. This bacterium is free-living, but is also enriched in genes involved in sugar transport and utilization and protein secretion, contributing to the cycling of carbohydrates and proteins within their hosts. There is even evidence that the evolution of Endozoicomonas has paralleled the evolution of its respective coral symbionts. And the bacterium Halobacteriovorax preys on common coral pathogens like Vibrio. Archaea of the clade Crenarchaeota are minor members of the coral hologenome that contribute to nitrogen recycling, and fungi seem to be involved in this process too. The protozoan Chromeravelia is a relative of the parasitic apicomplexans and exhibits frequent associations with coral, though the reason is unknown. Ciliates are common on corals, although they are mostly opportunistic hunters of coral-associated bacteria. And corals exhibit a high diversity of DNA and RNA viruses that probably contribute to nutrient recycling through their infections of other microbes. Corals, as mentioned earlier, can asexually divide or sexually produce new individuals, giving rise to massive colonies, barrier and fringing reefs, and atolls. These gigantic colonies, as we have seen, host countless microbes, but also provide rigid substrate for algal attachment. Urchins and gastropods can feed on the algae, and numerous other invertebrates such as sponges, anemones, starfish, and sea cucumbers, polychaete worms, bivalves, and crustaceans can recruit on the reefs too. These animals can be fed on by successive ecological tiers of fish, although tangs and unicorn fish will also eat algae. Large eels and groupers attend the reefs to allow small cleaners, whether gobies, wrasses, or shrimp, to remove their ectoparasites. And some fish have symbiotic relationships with anemones, such as the anemone fish that includes the well-known clownfish Amphiprion ocellaris. Corals are therefore the pioneer species that transform a nutrient-poor area into one of the most productive regions on Earth. But before we go, there is one more issue that warrants discussing: how these symbioses break down. It has been estimated that coral reefs are valued at over $20 trillion annually, so the recent coral bleaching and die-off events are a serious matter. Numerous studies have shown a link between global warming and ocean acidification on host corals expelling their symbionts, which provide the coral with up to 100% of its nutrition. The 2018 Communications Biology paper, Repeat Bleaching of a Central Pacific Coral Reef Over the Past 6 Decades by Hannah C. Barkley et al., found that the coral reef on Jarvis Island in the equatorial Pacific Ocean has historically recovered from bleaching events, but this recovery took time. The team noted that the Jarvis corals were not resistant to thermal stress, but the abrupt resumption of upwelling i.e., the wind-driven movement of cool, nutrient-rich waters, fueled rapid tissue biomass renewal in some species. The paper says, quote, finally, Montipora and Pocillipora species abundant on Jarvis prior to 2015 are fast-growing coral genera capable of quickly recolonizing a devastated reef following successful recruitment. Indeed, the dominance of these genera in Jarvis's highly productive, albeit relatively depauperate coral community may be a strategic trade-off that enhances the resilience of coral reefs in highly stressful environments, close quote. So we'll still have coral reefs for some time, but they'll have far fewer coral species on them than they previously did. But won't the corals just adapt to their new conditions? Some will, especially those that have been subjected to historic bleaching events. But that doesn't include most coral reefs. This is the conclusion reached by the 2018 PLOS Genetics paper, Potential and Limits for Rapid Genetic Adaptation to Warming in a Great Barrier Reef Coral by Michaela V. Matz et al., that points out, quote, we find that high migration rates do not disrupt local thermal adaptation, and that the resulting standing genetic variation should be sufficient to fuel rapid region-wide adaptation of Acropora millipora populations to gradual warming over the next 20 to 50 coral generations, or 100 to 250 years. Further adaptation based on novel mutations might also be possible, but this depends on the currently unknown genetic parameters underlying coral thermal tolerance and the rate of warming realized. Despite this capacity for adaptation, our model predicts that coral populations would become increasingly sensitive to random thermal fluctuations, such as El Niño Southern Oscillation cycles or heat waves, which corresponds well with the recent increase in frequency of catastrophic coral bleaching events, close quote. So, thanks for watching, and we'll see you all next time.